Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami microbiologist. Today in this video we are going to see another biochemical test, that is a salt tolerance test. In the previous video we talked about butyrate disc test. If you missed this, you can go back to my channel and watch the video. There will be also a link in the description box, that is the biochemical test playlist, so you will watch all the biochemical tests if you missed. Okay, so now it's time to talk about the salt tolerance test. So let's start the video. So in short form we can say STT. So again, this is the biochemical test in which we determine the bacteria. So those bacteria that have the ability to withstand or to tolerate a high salt concentration or to grow in a high salt concentration. Mostly we have the concentration 6 to 5%. Now remember, if the bacteria that are growing at a high salt concentration, we call that halo tolerant bacteria, or we also call that halophiles. Or those bacteria which are not growing at a high salt concentration, we call that non-halophiles. Now we have other terminology for the high salt concentration. We have the bacteria that are growing at a salt concentration. In salt we mostly use NaCl. So the bacteria that are growing in a high salt concentration, and that is the 1.7 to 4.8% or 0.3 to 0.7 m. These types of the bacteria are called as slightly halophiles. Okay, now those bacteria that are growing at a 4.8 to 20% or 0.7 to 3.4 m salt concentration, so we call that moderate halophiles. And finally those bacteria that are growing to 20 to 30% salt concentration, 3 to 4 or 5 to 9 m, so we call that extreme halophiles. Now we have other types of the non-halophiles that can also survive at a salt concentration. We have 2.5 m, so we also call that halotolerant bacteria. So these are the terminology that are really important to remember. Mostly these terminology are come in the MCQs in a competitive exam. Now in laboratory, mostly 6.5% salt are used to test the STT, means salt tolerance test. For salt tolerance test mostly we use 6.5% salt, or we can say NaCl. So due to this way we can easily differentiate Enterococcus species from the gamma hemolytic or non-hemolytic streptococcus species. So this is about the introduction. Now let's see the principle of this test. So we have the salt. Remember, salt is acting as a selective agent. Know why? Because the bacteria that are present in a mixture, let's suppose salt tolerant bacteria that are present in a mixture, so we can easily isolate that type of the bacteria. And we have membrane permeability and osmotic equilibrium. This is also possible by this salt, addition of the salt into a medium. So let's suppose if we have high salt concentration, so what will happen? Other type of the bacteria will be die and only salt tolerant bacteria will be grow. We have Enterococcus like. Okay, now what we will do to perform that test, mostly in laboratory we used BHI, brain heart infusion media. I already explained this media. Okay, you can go back to my channel and watch this. So we use BHI broth media that contain 6.5% salt, and we use NaCl. If we put this and we'll mix it and sterilize or autoclave it, so what will happen? Then we will inoculate our bacteria. Moreover, with BHI we also include some other ingredient like carbohydrate, dextrose, and color indicator like bromo, cerol, purple. So the addition of these ingredient with salt concentration, then we will inoculate our bacteria. Now after inoculation of the bacteria in this media, what will happen? The bacteria will utilize the sugar and the bacteria will survive in a high salt concentration, and we get and the bacteria will also release the product due to which the acid will be released and the media color will be changed. The pH will be dropped and the color will be changed from purple to yellow. This is how the test will be occur. Now if we see the procedure, so it's very simple. So we just take the bacterial colony, fresh bacterial colony, overnight fresh bacterial colony or 24-hour fresh bacterial colony, and we'll put it into the broth media that contain the salt as well as the ingredient which we already talked, means bromo, cerol, purple, carbohydrate, dextrose, this type of the ingredient. And after the incubation at 24 degrees Celsius, 24 hours, then we get the result. Sometimes the result will take 48 to 72 hours. Okay, so this is how the test will be performed. Now let's see the result. So we have the positive result. In positive result, what we have the turbidity in broth or without a color change from purple to yellow. This is the positive result. And in positive we have Enterococcus and Aerococcus species. These are the positive bacteria, STT positive bacteria. We have the negative result in which we have no turbidity, no color change after 72 hours incubation. So these are the negative color. And we have the bacteria like Streptococcus are the STT negative bacteria. Now these are the tube. As you can see, this is the uninoculated tube, which we just keep it as a negative control. This is the inoculated tube in which we just loaded our bacteria, but this is the negative result. There is no growth. Okay, now these two are the positive growth. This is actually BHI media, and this BHI media contains salt concentration. And as you can see, the color is you can see the turbidity as compared to this tube. So this is the positive result. Similarly, in this case we have also the positive and negative result. This is the positive result, as you can see. Now in this case the positive result is the yellow. So this is how we can get the result. Now let's see the uses of this media. So we mostly use this media again to differentiate the Enterococci from non-Enterococci. We also use this media to differentiate the non-beta hemolytic strain of catalase negative gram-positive cocci based on their ability to grow in a 6.5 sodium chloride broth. So there are some other species of the Aerococcus, like we have A. viridines and A. urini. It can also grow at a 6.5 NaCl concentration, means as a high salt concentration, when we keep it into the broth. So we can say that to differentiate Aerococcus species from other similar species like Pseudomatococcus and Helococcus. So to differentiate these bacteria from each other, we can also use this STT test. Now let's talk about the limitation of this test. So if you want to confirm the bacteria 100%, so remember, this is not a confirmatory test. For 100% confirmation, you may have to perform other biochemical test. So remember, sometime what we have other type of the beta hemolytic streptococcus like Streptococcus aglicete. It can also grow at a 6.5% NaCl concentration. So when it also grow at that sodium concentration, so what we get a confusion, because Enterococcus can also grow at that means salt concentration. So that's why other biochemical test is also important. There are some Enterococcus species or Aerococcus species that can produce a low amount of acid production, and due to this way we can also get a confusion in this result. Sometimes we have the bacteria which we talk that it not producing the acid, and these may be the species of the Aerococcus or Enterococcus, and due to this way what we get no change in the color, and due to this way it will give confusion. Now after the 72 hour we have to report the negative result. It is really important. It doesn't mean that 24 hour you see the negative result and you will report. Now after the 72 hour the result should be given. Then next we have to use light inoculum, because heavy inoculum can also cause turbidity and it will give the false positive result. Okay, so this is all about the salt tolerance test. I hope this video is helpful for you. Thank you so much for watching.